hay que compartir el enlace de YouTube para que nosotros lo podamos.
Pero ya lo tienen.
Si buscan el canal de Yul,
Das las gafas, por favor. Muchas gracias, Juan.
Tienen que ir a YouTube. Y en YouTube tienen que buscar el enlace de aquí de
La portada. Pero ¿para qué me estás diciendo eso?
las personas que lo van a ver desde YouTube.
¿Y ahora dónde hablo? ¿Aquí? ¿Cómo me oye? Entonces, haz atención, como hablo, no me hagas ruidos al lado. Vete un poquito más allá, porque se sienterá todo. Me pongo aquí, mira, así. Está bien, fenomenal. Entonces, ¿qué hora es, Alberto? La 1. Cierra la puerta, si puedes. Entonces, ¿podemos empezar? ¿Haces registración? Okay. Quizás se ve atrás de mí. Entonces, podemos empezar, ¿verdad?
Sí.
Okay, so I wanted to welcome everybody to the second International Winter Course and Workshop on Neuroimaging, the focus of which is on brain connectivity and mental health. So this is the second venue here in the Canary Islands in Tenerife. It's a Harvard Medical School collaboration with the University of La Laguna, and more specifically with the Institute of the University Institute of Neurosciences. And I wanted to thank all the colleagues here locally for the technical support and the organization they offered, and all the other colleagues that are online and participate in this venue. I hope in the future that several of you come here physically, not only to enjoy the weather during the winter, but also to start knowing each other and get into eventually fruitful collaborations. This is one of the missions of this conference. So the organizers basically are Stefano Pallanti for neuropsychiatry, Ron Kikinis for computer medical imaging, and myself for neuroimaging and neuroanatomy. And the local organizers are from the medical school, the histopathologist, Jose Luis Carrasco, the physiologist, the neurophysiologist, and imager of the university here, who is Jose Luis Gonzalez Mora, and the ex-dean of medicine and chief of neuroanatomy department, Agustin Castañeira Perdomo, and also Alberto Dominguez Martinez, who is the new director of the Institute of the University Institute of Neuroscience. Thank you all very much. So this is the mission actually in a nutshell, and the objectives of the winter course and this workshop on neuroimaging is to generate goodwill and forge quality international relationships. Here we are from Europe, and we are also from the United States, among colleagues that are motivated, basically that we feel enthusiasm for our work, and we want to see if there is a possibility to do quality projects in a way that we like it. Basically, a main issue is, do we like our colleagues to work with, which many of us with decades of experience in research, we know this is extremely important, the human relationships in research. So another objective is to see if there are ways to get funding from NIH, NIH institutions in the United States, but also here in Europe, and do collaborative grants. And of course, we know that our objective as educators and scientists is to elucidate and disseminate knowledge. So the issue of teaching students is very, very important. And here is a university, a vibrant university, and they really enjoy and participate in this experience. And also to achieve practical outcomes through the workshops, especially to use a very peculiar and special neuroimaging tool, which is the 3D slicer. So it's one of the few tools, or the unique tool in the field of neuroimaging, that can do manual work, and it is basically one of the favorite tools of neurosurgeons because it does surgical planning. It allows for very precise neuroanatomical delineation and anatomical delineation of any organ in the body using multimodal imaging techniques. This is the 4-day program, and today is the first day. And because of the time zone difference between the United States and Europe, we have a morning session that just finished here with the local people from the Institute of Neurosciences. And then at 1 o'clock local time, which is 8 o'clock in East Coast, USA, Boston area, for example, we start with our American colleagues. And today is basically a full day, you see here, based on neuroanatomy and some applications, important applications in different neuropsychiatric fields, like that we hear from Professor Fava, Maurizio Fava, in major depressive disorder and a major vision of psychiatry. Then David Perez with talking about neurological functional disorder and how he does his clinical research using multimodal neuroimaging and network analysis. And basically, after that will be Bruce Jenkins that will talk about neuroimaging, functional MRI, and structural MRI. Darren Doherty, who will talk about neuromodulation and especially in obsessive compulsive disorder with deep brain stimulation procedures. And last but not least, very basic talks in neurodevelopment from Pradeep Bide in Florida State University, and who carries on the tradition of Vern Cavines from Mass General Hospital, and he works with animal models. And two important lectures in neuroanatomy from Jared Rushmore and Doug Rosine from Boston University, that they will tell us really what it means from a neuroanatomical and histological approach, what it means, what we see with neuroimaging, structural neuroimaging. The second day, that is tomorrow, it will be, the leadership will be by Stefano Palanti, and he will talk mainly about neuromodulation and addiction. And it will be Stefano Palanti, Professor Henrietta Bowden-Jones from University College London, and John Camprodon from Harvard Medical School and Mass General Hospital. The following day, Saturday, it will be an entire morning on technological foundations of TMS. And there will be people that have developed this technology, like Risto Moniemi and his team from Aalto University with Victor Souza, Dubi Kicic, and Pantelis Liumis, and we will see the full technology and applications for especially with TMS EEG. So we will see how important is structure and targeting brain circuitry, identifying the brain circuitry with real-time tractography, brain tractography as you do TMS, and also getting the recordings from EEG. And so how do these correlate? And on Sunday, it will be basically an entire session on the use of 3D slicer for teaching anatomy. The workshop that goes in parallel is in a neighbor island in front of Tenerife, and it's Gran Canaria, Las Palmas. It is done in parallel with another venue, which is the NAMIC, and this is organized basically by Ronki Kinis and the local people here, Juan Alzola from the University of Las Palmas, Gran Canaria. And there are our techs from Boston that are teaching basically neuroanatomy using 3D slicer to technicians and students of our collaborators that are participating. Last but not least is the satellite seminar series that started today, and these are tailored for the Institute of Neuroscience at the ULL at the University of La Laguna. And this morning it was Juan Prieto, Pantelis Liumis, Stefano Palanti, and myself, who are also honorary members of that institute, of the Neuroscience Institute, and we talked about neuroimaging, TMS, EEG in the clinical practice of behavioral addictions, basically mainly gambling disorder. Tomorrow, there will be, for the same venue, the satellite seminar series in the early morning. It will be, which is really night in the United States, or very early morning, let's say. It will be, there will be presentations of the local people here, young students, postdocs, and doctorate candidates. So that is in the, let's say, an outline of this conference. And I will show you some achievements, some outcomes of last year's venue. And here you see the local colleagues in the medical school of La Laguna. And from right to left, the tallest guy is Jose Luis Mora, who is the neurophysiologist of the Faculty of Medicine and also the director of the imaging facility here in the hospital. It's the university and the hospital. The tower you see behind is the hospital, the clinics. And there is where is also the scanner. There are two scanners, Siemens scanners, 3 Tesla, for clinical and research use. Next to Jose Luis Mora is Jose Luis Carrasco, who is the histopathologist and the histologist of the Faculty of Medicine. And next to him is Agustin Castañeira, who used to be the dean of the faculty. He retired a few months ago, and he was the department chair, the anatomy department chair of the faculty here in medicine. And what you see, and on his side, it's me, and I have a little sample there of a tissue. Please be quiet. Okay, so the idea here was with these collaborations to work on the autonomic system, and basically what you see, it's a real cadaver that we got from the autopsy room in the Faculty of Medicine. Here they have a very large facility, one of the largest in Spain, of autopsies. They have 83 cadavers always in a very special facility with freezers that have different, and refrigerators that have different temperatures. So it is a wonderful facility to get tissue for any kind of medical research. And here you see an entire chain of ganglia for the orthosympathetic system. And in particular here, you can see the big one on the right side is the stellate ganglion. And that's what I have in my right hand in the tube. The idea was to get it in Boston, which we did, and to scan it. And here you see at the Martino Center, the 6.7 Tesla scanner for samples and mice. And with my colleagues, Bruce Jenkins and Andre van der Kuhe, we scanned it. And this was a preliminary result we got for such a small size tissue. And you can picture here the different sizes of and the scales for imaging. And on the right, you see at the 1 millimeter scale is what we do clinically commonly. From a 10 micra to 100 micra is the experimental research MRI. And then we are not yet at the nanoscale, but imaging research is getting towards, all right, the light microscope scale. And then we are going probably towards left, let's say, the nanoscale, who knows when, at the electron microscope. But this is where we go. So what we care a lot in all the research we're doing is the circuitries. And basically, it's this paradigm, and we'll talk a lot about that in the future lectures, of how the circuitries are considered biomarkers. And they're extremely important for psychiatric research and also for neuroimaging, because they can be imaged, they can be measured, and they can be quantified, and basically they can be monitored after treatment, and also by functional connectivity and structural connectivity. And this has been pointed out by Tom Insel 30 years ago or so, 25 years ago, and the work of Helen Mayberg, and it's known to most of you, all these achievements between 1995 and 2005, and that they focused our research basically and underscored the importance of studying brain connectivity for psychiatric clinical research. Basically, it's the paradigm of thinking of the disorders in psychiatry as disorders of connectivity, of brain connectivity. And the Human Connectome project is focused also on that, and it's one of the major goals of this entire project, how to really unconceal these brain networks functionally and structurally in the brain. And we use for that multimodal brain imaging with functional MRI, structural MRI, and MEG for the time domain, which is also extremely important. The behavior paradigm that emerged from modern neuroscience and this kind of considerations is what is called RDoC, Research Domain Criteria. Tom Insel was one of the promoters in the early 2000s, and it is a dimensional system for human behavior. And it is thought that it will make major breakthroughs in changing the paradigm really in neuropsychiatry, especially with the integration with the circuitry anatomy, detailed circuitry anatomy, physiology, specific paradigms of behavior, and experimental psychology. So getting all these different fields together in a quantitative manner by getting units of analysis like molecules, cells, circuits, physiology, specific behavior, self-reports, and paradigms. This is the objective of RDoC. And even if it seems as competitive to DSM, it's not. In my opinion, and in opinion of other colleagues like Stefano Pallanti, who will talk about these things tomorrow, they can be integrated, and it will be a great gain for both sides. So the collaborative achievements, just in closing of the first year, the last year's collaborations, were one paper that we just came out, let's say it was accepted yesterday, and it was with the anatomy and histopathology department here. And basically it was often from an idea of Agustin Castañeira, the anatomist, who observed that in several types of research, and especially COVID research, it may be extremely important, the study of the circumventricular organs, and especially three of these organs, which are the area postrema, the subfornical organ, and the organum vasculosum of the lamina terminalis. And this is because these are ports, gates of entry to the CSF in the brain because of the fenestrated and highly permeable capillaries in these areas. These particular regions are connected. These organs have neurons, and they project to other nuclei. So you can study, somebody can study the networks. Okay, and the importance that they have with the different modulations of autonomic systems, like, for example, the cardiovascular system, and also the brain-immune interactions. So this was one paper that came out of this collaboration. Another one that has been submitted and it is under review is a paper in investigating brain-immune interactions. Basically, this particular network, neural network, that can be acting on the dorsal dorsal vagal complex, basically the nucleus of tractus solitarius and the dorsal motor nucleus of the vagus at the areas of postrema, and it's an entire network that involves not only brainstem areas and nuclei, but also limbic system, the hypothalamus, and the cingulum, and other neocortical areas like the frontal cortex. So last but not least, two grant proposals, R01s, are in preparation. One has been submitted yesterday for February 5th deadline at NIMH, and it is to understand the pathology of the long COVID and its treatment. And the collaboration between Stefano Pallanti, Ron Kikinis, Zora Kikinis is the contact PI, and our group at Psychiatry Department in Mass General Hospital. And another project is the transcranial magnetic stimulation in gambling disorder, which gave a raise to an R21 with Stefano Pallanti, and we are moving, and John Camprodon, and we are moving towards an R01 with new data. So thank you very much. This is the introduction, the achievements, the missions, the mission of this conference. And this is Pantelis Liumis, a member of Risto Ilmoniemis group from Finland, who are the people that really will tell us how structure correlates with function by doing TMS EEG and then functional connectivity. Thank you very much. So I will take any questions now, and then if we don't have any, we can move to the next speaker. So let's see if there are any questions. And the next speaker will be Professor Pradeep Bide from Florida State University, and will talk on development neurobiology. Are there any questions? So Pradeep, you can start when you want.
Okay, let me. Are you able to see my slides? Can you hear me, Nikos?
Hay que poner voz.
Can you you can hear me and see my slides? Maybe not, Nikos?
Es que no puedo yo. Bueno, sí, sí, pues no.
Can you guys hear me or no? Can you hear me? Can you let me know if you can hear me or not? Nikos? I need some feedback. Can you guys see my slides? Nikos, can you see me or no? Oh, great. Okay, thank you. Thanks. So I'll assume you can see my slides and I'll begin. So my name is Pradeep Bhide. I'm at Florida State University College of Medicine. Let me just tell you, when I go to the presentation mode, I cannot see you or I cannot see anything else. So I will just continue here. I'm going to talk about developmental neurobiology, but particularly as it relates to developing preclinical models, which is basically animal models, to study neurodevelopmental disorders. As Dr. Makri said, neuroscience today, psychiatry particularly, is such an interchange between basic neuroscience, human neuroscience, and then clinical psychiatry. And I think animal models, cell-based systems make significant contribution to advance this knowledge from cells to animals to people to the clinic. But also, there's a lot of flow back from the clinic to the basic scientists or animal models or cell models. So I'm hoping that when I present my presentation, you will get a little bit of a understanding of how basic scientists who work with cells or preclinical models really benefit from knowing what the challenges are for human neuroscientists or clinicians. Understanding those challenges helps basic science to make their work so much more relevant than if they had been working on their own. So that's sort of my goal, tell you how we have used preclinical models to translate the findings to the clinic, or at least in a human neuroscience. All right, let me begin. What is developmental neuroscience? So depending upon who you speak to, it can be one of many things. It always begins very early in the embryonic period when the brain begins to emerge in the embryo. And many events occur: neurogenesis, production of neurons, neuronal migration, where neurons have to get to their proper destinations, and then differentiation of neurons, meaning production of dendrites, axons, synapses. So a lot of busy activities going on in the developing brain, and developmental neurobiology encompasses all of those things. For our work, we were interested in one particular thing, which is the role of neurotransmitters in brain development. For example, dopamine. So most of us know dopamine, glutamate, GABA neurotransmitters as chemical messengers at the synapse. So they're important for transmission of nerve impulses. But in the embryo, the developing brain, the fetal brain, there are no synapses. So the canonical function of neurotransmitters cannot occur in the embryonic brain. Yet, almost all neurotransmitters and their receptors appear very early in the embryonic brain, in the fetal brain. So these neurotransmitters and the machinery to transmit their signals is present well before synapses occur. So the question becomes, why are they there? Or rather, what are they doing in the embryonic brain? So we obviously, all the activities that are going on are neurogenesis, migration, differentiation. So the question is, do neurotransmitters have a role in these developmental activities before they begin their canonical function at the synapse? So what are they doing early on? To understand this, particularly with the focus on dopamine, we did a number of experiments. I'm showing you this slide to give you an idea of what types of studies we perform, what techniques we use. So we study neurogenesis, production of neurons using multiple markers. These red cells that you see here are the neurons that were produced over a 2-hour period. So we can delineate a small cohort. And just to show you, this is a control, and this is an embryo whose mother was exposed to cocaine. See a big difference here. Neuronal migration. Here, these green dots you see are migrating neurons. They are in this embryonic cerebral cortex of the mouse. Saline, cocaine. You see fewer neurons in the cocaine-exposed embryo. We can also grow these neurons in culture, and you can watch them migrate. And then there's another example of understanding dopamine receptor binding. So where does dopamine bind? Where are the receptors in the embryonic brain? This dark spot here is the striatum in the embryo. This is another example. And finally, we also do RNA sequencing or transcriptomic analysis or expression of genes in the brain, embryonic brain, adult brain. So we employ a number of technologies. And I hope this gives you an idea of what it is that can be done. Many of these things cannot be done in the human brain, obviously. So the animal models give a very unique ability to perform experiments at a cellular, molecular level that are difficult to do in human brains. All right, so what is it that I want to tell you about? So what we found, I told you before, dopamine is present in the embryonic brain. So it's doing something. In fact, we showed that it can modulate or influence neurogenesis, neuronal migration, synapse formation, gene expression. So the question was, what happens if there is dopamine imbalance? So normal dopamine levels are changed, the receptor is changed, and when might these imbalances occur in real life? So as Dr. Makris mentioned, one of our goals or one of our objectives was to study drug abuse during pregnancy. Whether it is cocaine, nicotine, alcohol, you can make a general statement that most addictive substances produce their effects ultimately by producing an imbalance in dopamine signaling. So I know that in the fetal brain, there is no addiction per se, but if a pregnant mother abused these drugs, dopamine levels in the embryo will change in the developing brain. The question was, what happens following this type of imbalance in the embryonic brain for dopamine signaling as a result of pregnant mothers abusing drugs? So we found, I know about 10, 15, 20 years of research, that this imbalance in the fetal brain, imbalance in the dopamine receptor signaling mechanisms, perturbs neuronal brain development significantly, and many of these imbalances occurring in the fetal brain produce persistent or lasting or long-term effects that can be manifested in the adult organism. Brain structure, behavior, all these things. So the question was, yeah, that's interesting, but does this really matter for us as as human beings who ultimately the focus of everything in the world? So with collaborators at Mass General, I used to work there many years ago. We found that if pregnant women smoked cigarettes, the risk for ADHD and other neurodevelopmental disorders in the children increased significantly. So cigarette smoking during pregnancy was an environmental risk factor for ADHD and other neurodevelopmental disorders. So here we were studying in the mouse embryo what happens when dopamine imbalance occurs. Nicotine, even though it targets nicotinic acetylcholine receptors, ultimately it does perturb dopamine receptor signaling. The reward pathways are activated and where dopamine plays a significant role. So our mouse model of nicotine exposure of a pregnant mouse and examining the effects on the offspring, the mouse offspring, could be relevant for human neurodevelopmental disorders that occur as a result of pregnant women smoking cigarettes. So we want to develop a mouse model, because ultimately the goal is to see if preclinical research could inform research in humans or in clinical scenarios. So before you can say, oh, we have a mouse model of any disorder, psychiatric, neurological, it needs to fulfill several criteria. And I'll go through these criteria and show you how we spent a lot of time making sure that our mouse model met these critical criteria, the three categories of criteria: construct, phase, and predictive validity. So what is construct validity? Basically means, is your mouse model constructed based on something that actually happens in the human condition, human situation? So as I showed you before, our mouse model of nicotine exposure during embryonic brain development recapitulates pregnant women smoking cigarettes. So that was fulfilled. In other words, nicotine exposure of the developing brain occurs in humans, and it is a significant risk factor for ADHD. So how do we develop a mouse model? Again, in one slide, I can tell you we did this and it worked, but it took about 10, 15 years of work. We experimented in many different ways, different strains of mice. So for those of you who don't use preclinical models, like everything else, each field has its own way of doing things, its own criteria, validation. So we use different strains of mice because strain of mouse can influence the outcome. Different periods of nicotine exposure, early pregnancy, throughout pregnancy, late weaning period. So basically we covered all 3 trimesters of human pregnancy as far as possible in a mouse model. Okay, so nicotine exposure, we use nicotine orally. We actually have now ability to expose mice to cigarette smoke, even e-cigarette vape or aerosol. We can expose pregnant mice to these different ways of nicotine exposure. So when we did all this, what happened? What were the behaviors? I will skip to behavior and not tell you much about the cellular molecular changes, because I think this may be a little bit more relevant to you. So these mice whose mothers were exposed to nicotine, so these mice were exposed during the prenatal and early brain developmental period, showed spontaneous locomotor activity, which is a proxy for hyperactivity, attention deficit, working memory deficit, and also impulsivity. So these are the major features or symptoms associated with ADHD. What about the neurotransmitters? Because those of you who study ADHD or many other psychiatric disorders, dopamine imbalance in frontal cortex, in the striatum, nucleus accumbens, the reward pathways, decision-making pathways, is a major contributor to many of these disorders, certainly to ADHD. Hypodopaminergic state in the frontal cortex of people with ADHD is considered a salient feature. The mice, prenatally nicotine-exposed mice, had a significant reduction in dopamine content in the frontal cortex. Not just the content, but dopamine release at the synapse, which we can measure in a mouse, was also reduced. So behavioral changes, hypodopaminergic state. What about neuroanatomy? So Dr. Macris and his colleagues had shown that in ADHD, untreated ADHD subjects, there are structural changes in the cingulate cortex, cerebellum, and other areas. Quite remarkably, we weren't expecting this. When we did diffusion tensor imaging in our mouse model, prenatally nicotine exposed, we saw microstructural integrity was reduced in the frontal cortex. This is a mouse brain, medial prefrontal, let's say, frontal cortex, dorsal striatum, and also in the cerebellum. And we were quite struck by how well it recapitulated the human ADHD in terms of neuroanatomy, in addition to behavior, neurotransmitter, etc. So this is called face validity, meaning does your model look like the disease that you're trying to model? And I think the answer is yes, behavioral, neuroanatomy, dopamine imbalance. And the final criteria is predictive validity. Can you use a drug and can you get a response in your model that will predict its efficacy? So a little convoluted way of saying it, but basically the idea was if we gave this mouse, which shows all these symptoms, neuroanatomical changes consistent with ADHD, will those things change? Will the behavior improve? Will dopamine go up in the brain? So we used this stimulant medication, methylphenidate, or Ritalin, or Focalin, and we gave that drug to this mouse model. Again, took quite a bit of care to make sure we were giving a dose that will be equivalent to the therapeutic dose in ADHD patients. It's never going to be exact. It's always going to be an extrapolation and estimation, but we didn't give too much methylphenidate. But when we administered methylphenidate to this mouse, a single dose, we gave oral or systemic, didn't matter. The hyperactivity was reduced, attention improved, working memory improved, impulsivity reduced, and when we measured dopamine release in the frontal cortex, immediately upon administering methylphenidate, dopamine release went up. So the behavioral changes had a basis in neurotransmitter changes. You couldn't do this type of experiment in the human subjects, of course. So the mouse model had a construct validity. It was developed based on an observation in human literature that smoking during pregnancy increases the risk for ADHD. The mouse model showed the behavioral symptoms, neurotransmitter symptoms, neuroanatomy, and it responded to stimulant medications. So now that you have this mouse model, although it took a very long time, what do you do with that? And this is where I think I will give this example, because if you are thinking about this is a discussion rather than a lecture. So if you're thinking about a particular phenomenon that you have observed, or a disorder, or a particular treatment modality, and you want to perform rigorous hypothesis-driven, well-controlled experiments that you cannot easily do in humans, you can use a mouse model, validate the mouse model using all these criteria, and then you can advance your research or understanding. So what did we do? I'll just give you one example. We used this mouse model to screen novel compounds, or at least previously unknown potential candidates for treatment of ADHD. And we discovered that a kappa opioid receptor antagonist, that's a long name, norbenaltorphamine, was a novel non-stimulant candidate drug for treatment of ADHD, as well as another neurodevelopmental disorder, a rare disorder, Fragile X syndrome. Company to take this drug through the preclinical, clinical, and ultimately phase 2. We have not gotten very far, unfortunately. We are still in preclinical stages. But an example of how a preclinical model can help discover and even advance novel medications for the benefit of society. And this is a collaboration with multiple clinicians at Mass General, as well as scientists here at Florida State and University of Florida, etc. So I'm making these sweeping statements with one slide representing many years of work. So that's a direct example of a preclinical model helping discover new treatment opportunities. What about research? Can you advance research using this animal model? Something that you cannot do in people, and also something novel, pushing the frontiers of neuroscience, developmental neuroscience. So here's an example I'll give you, but I'll take you back a little bit, give you a little story here. Many of you know that there is this Dutch famine, or the hunger winter of 44-45. Towards the end of World War, northern Holland, the Netherlands, suffered a pretty severe famine. Millions of people suffered. But what's interesting is today, or let's say in the last decade or 15 years, papers were coming out showing that grandchildren of women who were pregnant during the famine have physical and metabolic traits that are not seen in any other population in the Holland. So selectively, 2 generations later, symptoms are appearing, metabolic and even physical characteristics like height, that can be traced back to that famine. So this observation and many other observations from human research led to a new field in neuroscience or in biology. It's called heritable influences of environmental factors. So environment-induced traits that are heritable, that are shown in multiple generations descending from the directly exposed generation. Famine was one example, but of course we all know that in the 50s and 60s, tobacco, cigarette smoking was very prominent. In fact, as you can see here, in some cases, physicians would recommend cigarettes to pregnant women for anxiety, morning sickness, etc. So highly prevalent environmental factor in the 50s and 60s. And the children born to the women who smoked today are adults, obviously. So our question was, if famine can produce traits that linger in the population for generations, can smoking during pregnancy produce changes that can show up in multiple generations, even though only the mother smoked? Children never smoked, grandchildren never smoked. So that's the experiment. A lot easier to do in a mouse model, as you can, of course, imagine. So we did that experiment. We had already our model. So pregnant mice were exposed to nicotine. We bred these pregnant mice with unexposed male mice. So the father was never exposed. We got the children. We bred them one more time with unexposed males or females. We got the grandchildren, and we went one more generation. What we found is that the children, of course, I showed you, they have all the ADHD phenotypes. All the data I showed you were from children born to pregnant females who were exposed to nicotine. But we found that the grandchildren and great-grandchildren, so 3 generations descending from this female mouse exposed to nicotine during pregnancy, showed hyperactivity. So the hyperactivity phenotype was transgenerationally transmitted. Then we also realized that not only, psychiatrists, particularly in olden days, would blame the mother for everything, but really men smoked cigarettes as well. In fact, more men smoke cigarettes then and now than women. The question becomes, can smoking cigarettes by the father produce traits in children, grandchildren, due to this transgenerational transmission? So in fact, with our colleagues at MGH, Dr. Biederman and Tom Spencer and Steve Ferrone, they analyzed their databases, and there was a very preliminary but significant link between father smoking and children developing ADHD-like symptoms, even when the mother did not smoke. and smoking around the time of conception of the child. So went back to our animal model, and we exposed male mice to nicotine, not pregnant mice, it's male mice. We bred the male nicotine-exposed mouse with a unexposed female, produced children, one more time, produced grandchildren. And we did various behavioral analyses in these two generations, children and grandchildren. Remember, they were never directly exposed. And we found hyperactivity, attention deficit, cognitive inflexibility, as well as changes in gene expression in the brain in these two, got it, yeah, in these two generations of animals. So one of the advantages of animal models is you can try to understand the mechanisms of this transmission. And we found out that it's the germ cells, the sperm of the male mice exposed to nicotine that had undergone epigenetic changes in DNA methylation, microRNA expression, nucleosome repositioning, etc. Those changes in the sperm somehow were responsible for transmitting the molecular, genetic, and behavioral changes to multiple generations of offspring. So just to mention, we also recently found that the artificial sweetener aspartame produces anxiety, not attention deficit, not hyperactivity, but anxiety in multiple generations. And we showed this with learning and memory deficits, anxiety, and changes in gene expression in the amygdala. So it's not only nicotine, but other environmental causes can also produce these changes that can linger in the population for multiple generations. This, I believe, has significant implications for public health, epidemiology, regulatory science like the FDA, for example, and also in biology, evolution itself. So I want to tell you that this animal model that we developed gave us some advantages for drug development, as well as advancing science in ways that we could not have done by using human research alone. I just want to tell you what's going on right now. We are continuing all this work. We are looking at various mechanisms. We're focusing on Fragile X syndrome, multiple technologies we're using. So with that, I'm going to stop and let you know that I hope this hopefully helped you to think about how preclinical models can help advance your research or research for anyone, and have multiple, hopefully potentially significant implications. Thank you, and I will take any questions if there are. I hope I stayed on time. I don't think I can hear you for some reason, but I'll...
Pradeep, can you hear?
Oh, yes, I can, yes.
Thank you so much about this wonderful talk. Also, you brought us back to the John Biedermann's day.
Yeah.
So I have one quick question, and I will ask.
I can't hear you. I can't hear. I cannot.
Okay.
Yeah, no, I can.
Quick question. There is a problem with the sound.
Yeah. Oh, it's okay now.
Okay. At least let him listen to you, and then I'll.
Okay, so you can hear.
Yeah.
So basically, my question is the following. Remember the big issue with Joe that and the debate, what would be the reasonable and safe age to give medications to children? with ADHD, and could be with other pathologies also.
Right, right.
So,
So
With
if you look at
sense, the last 10, 15 years, if there have been established some rules for this kind of medications from translational studies, has something emerged that is important for us to know.
yeah, so in our research, I think what happens is in the preclinical research, sometimes people don't calibrate the dose of the medication that they're testing based on what is therapeutically equivalent to the human dose. So when you administer stimulants to animal models at doses that are representative of clinically therapeutic doses in humans, we don't see any changes in brain development. That's the only thing we studied, behavior. However, there are many papers using rat and mouse models that show potential effects on the developing brain, but they have used very high doses of stimulants. So as far, and even with the safety and toxicity studies, therapeutic equivalent stimulant medications have no adverse effect on the developing brain. What you're asking me, what might be the safe age to, or is it safer in young developing brains? Preclinical studies suggest that therapeutic doses they don't produce toxic or adverse structural or functional effects. That answer your question?
I wonder whether, for example, like in neuromodulation, we use neuromodulation and we don't know exactly the specificity of the circuits we are targeting. But we do correlational studies, but we have also imaging techniques like functional imaging and functional connectivity where you can see plasticity in a certain way, a plastic response.
Mhm.
I wonder if there are studies like that can be done in early age and can help understand the effects of drugs.
Yeah, there are multiple ways to do that study. And one of the common ways is there are these genes called immediate early genes that are expressed as soon as a particular pathway is activated or stimulated. And in mouse models, a simple example, if you play a sound or you flash a light, the visual or auditory pathways, or if you stimulate a whisker, they get activated. And you can perform studies in brains post-mortem in an animal and identify cells, structures, and pathways that are activated, sort of like your fMRI. That's one way. Or there are markers for synaptic activity, calcium imaging. There are multiple ways that you can see if any stimulation that you do produces immediate effect, or if you keep doing it repeatedly, does it produce long-term effects and change the circuitry? It is possible to do those studies.
Yeah, thank you very much, Pradeep.
Okay.
Thank you. So we are going to our next presenter, and this is it you, Doug, or Jared? Is it Doug? I think it's Jared.
It is.
It is Jared.
Jared.
Yeah, I mean, it depends on you too who wants to be next. I don't know.
I'll go.
Okay, so you're using Zoom.
Well, why don't I try with the Google business, and then if that doesn't work, I can go either way.
So why don't you go to Google Stream?
Okay, I will.
So he will go with Google Stream.
Okay.
Okay, just give me a second, please.
Yeah, take your time because they are finagling something here with the monitor.
Or,
Google Stream.
Yeah, I think we're ready now.
yeah.
Anytime you want, Jared.
Okay. Okay. Can people hear me and see me?
Yes.
Okay. Okay, well, good afternoon, good morning, depending on where you are. I'm Jarrett Rushmore, and I'm absolutely delighted to be here. In fact, I'd rather be where you are than here, because here is Boston, which is around 30 degrees Fahrenheit, and windy and cold and snowy. But in any event, I can live vicariously through you. I'd like to first thank Dr. Makris and the organizing team for this course, and I'll present my presentation entitled The Importance of Anatomical and Histological Validation in Neuroimaging. Now today I'm going to talk to you about what we know directly about the neuroanatomy of brain connections. Now the word directly is an important one here, because one of the things I hope to convince you of here today is that there is very little precise and direct knowledge of human neuroanatomical connections in the scientific literature. And when I say direct, I mean the knowledge of where a brain pathway begins, where it travels inside the brain, and where it ends. Now, we have several indirect ways of delineating connections between regions in the human brain, but these methods produce connectional data that may not be always accurate. Thus, if we don't have the knowledge of brain connections in the human, then how do we judge the accuracy and the precision of methods that indirectly show connections in the human brain? Now, in some respects, this represents a very central problem in understanding how regions of the human brain are connected. And today I'm going to tell you about how we reached an understanding of this issue by asking a simple question. That is, where are the data and the methods underlying direct visualization of connections in the human brain emerging from? We approach this in a slightly unconventional way, as I'll tell you in a few minutes. I'll then give you an idea about how we can fill this gap in understanding and improve our ability to accurately measure connections in the human brain. Okay, well, we'll start the story when I was starting out as a faculty member here in the Department of Anatomy and Neurobiology. And I started teaching a course in advanced neuroanatomy. I had been teaching medical histology and neuroanatomy for many years, and I'd been burning details of spinal cord and brainstem connections into my students, much to their dismay, I should say. And when I started developing this graduate course, I wanted to dig into the science behind the Gray's anatomy and the textbooks that we teach in medical school to understand really where the source of information was from these pathway tracings. Part of this was that the textbooks told me that there was a large number of spinal cord tracts, for instance, only a fraction that were present in the medical school curriculum, and I wanted to know about every one of them. I wanted to get a comprehensive understanding of these pathways. So I dug into the literature, and as I did, I found that many of the fundamental papers detailing, for instance, spinal cord anatomy, but other pathways, were not in the human. And in fact, most of them were not even in a primate. A lot of the foundational knowledge appeared to be from species such as the cat and the rodent. And anybody who has a cat understands that their movement repertoire and abilities are considerably different than that of a human. And you can surmise that the connectional neuroanatomy that connects the spinal cord with the brain may be similarly different. So later on, I sort of got into an idea with Dr. Makris on exploring how extensive this was. So we sat down with the first classical neuroanatomy text, Malcolm Carpenter, from 1983. And this is a foundational text. And I went through all of the references and I pulled out all the journal articles and determined what species they were from. And the question here is, where is human neuroanatomical knowledge coming from? And over 2,000 primary references were examined. And here's the results of what we found. We found that in this textbook, almost 37% of all of the studies were studies done in the feline. And only a minority, only around 13% of the studies in this so-called human neuroanatomy book were derived from studies in humans. Well, this was sort of interesting. Then we dug into what was the studies in humans, and we found that these weren't really connectional studies at all. These didn't detail how one brain area interacted with another brain area. Now, these were studies that looked at things like blood-brain barrier, blood-brain vessel, neurological syndromes, and gross anatomy and embryology. So the connectional studies were surprisingly limited in this particular textbook. And then we sort of asked the question, well, this was a 1983 classic textbook. Let's go to a more modern textbook and see if that recapitulated this understanding. And so we went to the famous Nieuwenhuis book, The Human Central Nervous System, a brilliant and very wide-reaching book. But when we did this, we evaluated around 3,400 references and just asked the same question of what species were being used in the evaluation of the data. We found a similar trend. That is to say that a minority of studies, and very similar in terms of anatomy and embryology and blood vessel architecture, these were present in this book, very few connectional neuroanatomical studies in the human. Now, I'll also note that there was a difference in the proportion. We had a 37% in the previous textbook that were devoted from cat. And here we can see a reduction in the studies that were cited from the cat and an increase in the number of studies cited in the monkey and in the rodent. And this reflects, I think, a general change in the field as we move from feline models to more primate and rodent models. But in any event, we found a similar trend that a minority of studies were in the human. The nice thing about this textbook is that it allowed us to evaluate these data according to different systems. And so in, let's say, the limbic system, we could identify that the rat, or the rodent, I should say, made up much of the content of these foundational information, as did the reticular formation and things like the hypothalamus and ventral thalamus. In other species, such as the motor system, the cat still was a big player, as was the macaque. And so different species, in fact, combinations of species, were pulled together to derive the understanding of neuroanatomy for each of these neural systems. And I'd just like to point out this figure over here, in which we see a cross-section of the mid-sagittal section, I should say, a cartoon of the human brainstem, diencephalon, here. And what we see is that the connections that are overlaid on this human cartoon are actually derived from the rat. And so it's not uncommon in this textbook and in other textbooks to have the information from a rat or a cat or a primate model generalized to the human. So what I want to convey is that of the studies that we've looked at, that a small proportion of them were in humans, and a small proportion of those were in fact connectional studies. So why is that the case? Well, it's the case because of ethical reasons. To really elaborate what we mean by connections between one brain area and the other brain area, we need to understand three parts. We need to understand the location of the neuron, the location and the topography of its axon, or what we call the stem, and where the pathway ends, or the terminations. And this is really only possible to directly visualize all three of these aspects if you do invasive neuroanatomical tracer studies. And over here we see an example where we inject a substance near the cell body. It is taken up by the neuron and transported down the axon. And in doing so, we're able to see where the location of the cell bodies are. We can see the axons, and we can see the terminations. And so you can see an example of that right here, where there's a tracer injection into this part of the macaque monkey brain, and you can identify the axons that are streaming away from this towards the corpus callosum here. A similar approach is called the retrograde neuroanatomical approach, where you inject in the vicinity of the terminals, and the tracer is then transported retrogradely back to the neural cell body to identify the neuron cell bodies that give rise to the projections in the injected area. And so for these reasons, the only way at this present time that we can examine all three of these components of the pathway are by invasive neuroanatomical studies in animal models. Well, that being said, I mentioned to you that some studies, connectional studies, were done in the humans. And what are these? Well, here's some examples of the methods that are used to elaborate connections in the human brain. On the left-hand side is a dissection method called the Klingler method, in which the brain is frozen and then fixed, frozen and fixed in cycles, making the white matter very amenable to micro dissection. And these are two examples from my laboratory where we examined the micro dissection approach, the corpus callosum, and of the visual system here. So this will allow us to see the stems and infer to some degree of accuracy where the terminations are and where the origins are, but we cannot visualize the terminations per se. We cannot visualize the branchings of the axons, and we cannot visualize the cells of where these little string cheese bundles actually find themselves. A classical way of evaluating connectional neuroanatomy is through histology. And the most famous folks are the Desjardins from the late 1800s. And these folks took very carefully stained sections of the human brain and examined with a lot of precision the different fiber tracks within the white matter of the human brain. The issue here and the gap in knowledge is that just like the microdissection method, this did not allow for an evaluation of where the fibers were coming from or where they were going, but it allowed an understanding of the organization of the fibers within the white matter. There's also a body of knowledge that looks at degeneration. That is to say, when a human has a stroke, there is degenerative changes that occur in the spinal cord, in this case, that allow you to understand to some approximation the pathway that is affected by the stroke in long distance. However, this is a bit this is very broad because you can get lots of information that are false positives as the lesion increases or decreases, or secondary effects of degeneration. There are methods that use tracers applied to the dead brain, and this is a slide from Dr. Goluska in Germany, which applies a lipophilic dye to a piece of dead brain, and then over the course of 6 months or so, there is a limited transport of that through the plasma membrane to illuminate certain small connections. But by far the most prevalent and promising and amazing method, I should say, of evaluating connections in the human brain are diffusion MRI-based methods, as we see here. And this has, I think, completely revolutionized the field of neuroanatomy. But that being said, this method suffers from many of the same issues as the dissection method and the histology method. This is to say, it is very difficult to identify all the stems, where they're located, where they're coming from, and where they're going, the axons and the pathways. And so these are inferences based on the diffusion of water, and we'll get to those in a bit. I also would like to mention that this method of microdissection, which I illustrated here, is undergoing an amazing resurgence as people are realizing the absence of gold standard information about connections. They're going into more and more detail about the type of information that can be obtained from the brain dissection approaches. And so here we're starting to see papers coming out that detail the very precise organization using microdissection. Here you can see a small tweezer peeling away a bundle of axon in the temporal lobe. We're also seeing folks that are integrating the microdissection approach with a contemporaneous ex vivo MRI to try to merge information from different fields and thereby produce a higher resolution of understanding. But even so, this is still going to be subject to some extent to the limitations previously discussed. So what I'd like to present to you today is an ancillary approach, a separate and parallel kind of way to get at the gold standard information of what brain area is connected to another brain area. And to do this, what I'd like to illustrate is this figure, where on the left, we see what is known in the macaque monkey, in the model. Remember, the macaque monkey allows us to evaluate the origin of neurons, such as this, where the neuron sends its axon in the white matter. And this can be done by track tracing and complemented by things like microdissection and diffusion MRI techniques. And we can identify the termination pattern. In the human, by contrast, we can identify the fiber stems, as the Desjardins or diffusion MRI and other things have demonstrated, but we cannot evaluate the origin or the termination of these fiber pathways. So how do we use this? How do we leverage the macaque monkey to inform on the human? Well, I'm going to argue that using the methods of experimental track tracing in the monkey and extending that by extrapolation to the human provides a way to validate connections in the human brain. At this point, we can evaluate thousands of experimental track tracing cases, which have been done over the course of many years and the course of many laboratories. And these have generally been collected and collated into connectional databases that look at specific regions and where it connects. And this is a picture from Marcus Kaiser in 2017 showing the results of that connectional database and how it maps onto a lateral view of the brain. We can use these to generate network figures and network parameters and outcomes to show the connectional organization of the brain. And we can then use that to go to the human. And this has been done to some extent. Here is a paper by Catani and Thibault de Schouten in 2012 that showed very close alignment of many of the major fiber bundles in the white matter that is both in the monkey and the human. And so there is a connection, there is a homology and an extrapolation that can be done. Now, we should say, I think very concretely, that this does have limitations. We can extend what we know from the monkey into the human, but they are two different species. The monkey, we can predict that things like the visual system and the limbic system are likely to be very similar, but the monkey has a more limited behavior repertoire than the human. And so the pathways that underlie this are expected to be different. So we have to be targeted in what we do. But fundamentally, we feel that there is a foundation of connectional neuroanatomy in the human that at a gold standard level that can be extended, in the monkey that can be extended to the human. And the way that we intend to do this is by using an analog of the famous Harvard-Oxford Atlas that was developed at the emergence of the MRI field by Dr. Makris and others, and extending that to the monkey. And here we have an example of an MRI, an ex vivo MRI that's been parcellated of the monkey brain according to a modified Harvard-Oxford Atlas system. And this allows us to take parcellation units or sectors of the MRI brain in the monkey and look at them in terms of the analog in the human. And presumably this will allow the foundation for us structurally to take connectional information from the monkey and overlay it to connectional information in the human by having a human and macaque parallel system that are linked through cytoarchitectonics and landmarks. And here's an example of how we can use this. This is the same Harvard-Oxford Atlas from the macaque overlaid here in transparency. And what we can do is scan this brain with diffusion MRI and take a region of interest in the frontal cortex as such here. We can then use this region of interest to produce pathways that we can reasonably view in the diffusion MRI data, something along these lines. And then we can ask the question, does this pathway results that we see from diffusion equate to what we see in histology? And to do that, what we can do is take advantage of and leverage what we're referring to as the Pandya-Rosene Neuroanatomical Archive here at Boston University. This is a collection of over 600 individual neuroanatomical track tracing cases, section by section by section, and they look something along these lines. So we can see the ground truth information of connectional neuroanatomy here. We can then take the region of interest and equate that to the injection site, which we would see in this particular case, and then compare the results from the diffusion directly to the results of the neuroanatomical tracing in a different animal. And in doing so, what we can do is something along these lines. We can take this injection and say, we expect, based on the anatomy that we see in our slides, that this region is connected to the following parcellation units in the Macaque Harvard Oxford Atlas. And then we can evaluate different types of diffusion models and see how well they do against the ground truth. We can use that to help improve the diffusion atlases and algorithms and more closely align them with the results from the neuroanatomical tract tracing. But that being said, I think that approach being said, I think that the state of ground truth in the macaque can also be considerably improved. So if we take a look at the studies that have been done in this COCO-MAC database, the gold standard of connectional databases in the primate, we see a couple things. We see that if we go back to around 500 or 450 or so manuscripts, and we look through the manuscripts and ask the simple question of, is there information about where the axons are, we find a surprising result. The first thing that we find is that most of these studies use retrograde tracers, the vast majority of which just identify cells. And here's an example from Dr. Moorcraft where there's an injection in the cingulate cortex, and we see the cells that give rise to the axonal terminus in this region. But what we do not see is the axon pathways that link these. And so we don't know, based on these data, whether the axons go through the gray matter, whether they go through the white matter, or whether they go through both. And we also found in looking at these that most studies using anterograde tracers, where you inject the tracer, it's taken up by the cell body, and then it projects to its terminus, these only report the terminations and not the location of the white matter pathway in the white matter, in the cerebral cortex. And it turns out that if we sort of figure this out and run the numbers on the back of an envelope, very few of the manuscripts, and we're estimating around 2% of the COCO-MAC database, very few of them report information on the white matter topography of the labeled connections in the cerebral cortex. And here you can see the sort of dramatic effect of this. So what we have is very good information on where a pathway starts and where a pathway ends. But what we don't have in all cases is where that pathway is in the white matter. Now, there are a few very important papers, and in fact, one incredibly important book that are attacking this and have attacked this. But I want to convince you that a huge amount needs to be done to really evaluate the topography issue in the macaque monkey. And I'm also here to sort of tell you that this Pandia Rosine Neuroanatomical Archive that I curate is a potential source of examining this, because we have a lot of these data, but we also have a lot of the data that shows these pathways, which were not reported in the original data. And again, we have around 600 or 650 cases that we can examine. Now, why is all of this important? Well, it's important because the more we know and learn about neuroanatomical connections, the more we understand that that the axons do not take just the shortest path to get to where they want to go. So here we see that axons are complicated. They have branches. They do not go in a linear way to their terminations. And here in this paper from 2021 is a wonderful example of this. Here there was an injection into the thalamic region, which would be located here a couple slides back. And we are looking at the axonal terminations of that anterograde injection. And what we're going to do is we're going to look at a three-dimensional reconstruction of this sulcus here and find that the axons in this panel on the far right are coming in. They're going up this gyrus. They're making a hairpin turn to curve around to reach their termination point. So they don't go the shortest path from here to here. They take a very unusual path. And in fact, if you look carefully, you can find that many of the axons actually enter the gray matter instead of turning around in the white matter and go through the gray matter of the flanking regions of the sulcus to get to their termination. So all of this is to say that we need this information because biology and axons are complicated. You can also see that here. And I want to sort of also emphasize in this section that there does also seem to be a layer of organization to this. So it's not just willy-nilly random spaghetti. There does seem to be unusual trajectories, but there also seems to be some order to it. And you can see this in these two cases. Here's one case on this row and the second case on this row. And what you see is an injection here and an injection on the other side of the bank. You can see that this injection here produces a fiber bundle that goes underneath the cortex here and then enters the cortex here. And the other way around, when we inject here, it kind of avoids that area and sends its lighter projection at a lower substrata. And you can also see that when they're both projecting to a similar place here in the cingulate sulcus on the dorsal bank of this sulcus, the dorsal, this case right here, comes up and around here underneath the white matter junction in the gray matter, where this case maintains its position in the gray matter, in the white matter here before going up to terminate there.
So you mean?
Okay. This is my last slide, or one of the last slides. And what I want to convince you of here before Dr. Rosine takes it to the next level is what we need for better diffusion MRI, for better human neuroanatomical connectional neuroanatomy understanding, is more work to be done in relating experimental track tracing methods to diffusion imaging. And I'm happy to say that more and more work is being done on this as people start to recognize that the gold standard information that's present in the tracing studies of the macaque monkey can now be applied to inform the results of the MRI and to improve that. So what I've hoped to convince you of today is that much of what we think of foundational human neuroanatomy is actually derived, particularly when we think about connectivity, from work in animals. And while this is not completely awful, and especially with analogies that are present in the macaque monkey and the human, it may be difficult to relay those information for species like the cat and the rat. So these this limited information is derived from both the methods that are possible right now and that prompts us to advocate for the use of extrapolation methods from the macaque monkey ground truth information to extend, validate, and confirm the results of diffusion MRI to improve connectivity information in the human brain. And with that, I will stop the share and invite any questions.
Yes, there's a question. So, Jared, thank you so much for this masterful presentation. So I will have several questions. Yeah, one critical question is, on the light of all this evidence that you are showing and your experience, how specific can it be a target for a circuitry in the dorsolateral prefrontal in the human?
I'm sorry, what was that? How specific?
Yeah, how specific? How accurate and specific can be our evaluation of a circuitry when we target some area of the dorsolateral prefrontal cortex with TMS in human?
Well, I think that it's going to be hard to say, Dr. Makris. I think that we have to not only rely on neuroanatomy for this, but we can also rely on physiological correlates of the neuroanatomy that can be obtained with TMS. We can look at changes in behavior. We can look at other things that are specific for dorsolateral prefrontal cortex. In terms of what I think you might be getting at, how well can we extrapolate from the macaque monkey to the human, given the differences in size of the dorsolateral prefrontal cortex and knowing the ground truth information, I think we can do a pretty good job. But I don't think that we can precisely evaluate the connections of the human brain in the dorsolateral prefrontal cortex from the monkey. But I think we can use it as a foundation and to improve the results from diffusion MRI.
Thank you so much, Jared. So we'll move to, because of the shortness of time, we need to be precise for the next presenters. We'll move to Doug Gerzin, Professor Doug Gerzin, but we'll follow up on what Jared has started already. Please, Doug, go ahead.
Okay, I'm trying to bring my presentation up, and this will let you take over Jarrett Rushmore's main presenter. Okay, window on my computer. This is sharing. And okay, can you see the first slide and hear me, everybody? No? Yes? Maybe? Nikos, can you hear me and see the slide?
Yes, I can hear it very well and see it very clearly.
I'm not sure what's going on. I'm not getting a response from anybody about my slide or my window.
Yeah, we see your window. You have 8 slides there. You have 6 slides.
I can't understand what you just said, Nikos. It was garbled.
Let's use this one.
Okay.
Can you try
You want me to try to share again?
no audio here?
Can you hear me, Doug?
Yes, I can hear you.
Okay,
Can you
we
hear
can
me?
see your slide. We will see your slides and hear you very well.
You can. Everything's okay?
Yes.
All right, so let me try to share again.
We cannot see them anymore. You can't hear?
Yes, but the slides is not presentation.
Now you have to put the slices on again. They went away.
All right, it now says I'm sharing. Can you see?
Okay now?
I'm okay?
Yes.
All right, fine. So I'm going to talk about neuroanatomical assessment of MRI tractography, specifically diffusion MRI, using track tracing, as Jarrett just explained, and some immunohistochemistry, but to ask a little different question. That is, in diffusion MRI, what are we really seeing? What can it show? And this was done in collaboration with Farzad Mortazavi, who's with me at and Jarrett at Anatomy and Neurobiology at BU, and my colleagues Van Woodine and Jeremy Schmaman at MGH. And big thanks to Nikos and the organizers for inviting me to do this. And like Jarrett, I wish I was in the Canary Islands instead of here, but maybe next year. Okay, so this is an example from a book by Terry Williams of what you can do with that Klinger method, as Jarrett just showed you. That is sort of the simplest kind of connectional anatomy. This is a myelin stain, and you can see these fiber bundles, but you can't trace individual fibers, even in the most sensitive myelin stain, which is shown here, black gold, in a rat brain. You can, however, see areas of degeneration if there were a stroke, say up here in motor cortex. And this has been the mainstay of connectional neuroanatomy until the 1970s, when radioactively labeled amino acids were developed. And with Jeremy Schmaman and Dee Pandya and Gary Van Hoosen, I immediately began making injections into monkey brain. And with Dee and others, that's where this big archive came from. Now, Dee and Jeremy took a lot of this material and identified things like the cingulum bundle, the frontal occipital fasciculus, but couldn't really follow them. But Van Woodine at MGH developed a very high resolution diffusion method that he refers to as diffusion spectrum imaging. And he too then saw things like the frontal occipital fasciculus, cingulum bundle, and identified these, but was still left with where do they start and where do they end, as well as exactly how do they pass through this huge area of white matter. Now, in 2012, Van published this paper, and I was honored to be on it, looking at his diffusion spectrum imaging in the rhesus monkey, owl monkey, marmoset, galago, and he had similar human material. And we were all puzzled to find that while some of these fiber pathways curve along the way you expect, there were a lot of what Van identified as grids, areas where the fibers were going in XY directions. And here, in fact, at higher power is an example of the SLF corpus callosum crossing, okay, and forming a two-dimensional grid, obviously, and then there's a third dimension coming out of it in some places. And here's another example. This is rhesus monkey, where you can find grids in the central sulcus, in the corpus callosum, and in the arcuate sulcus. So it seemed like these were ubiquitous, and this, in fact, is how the brain develops. In the early vertebrate brain, things follow X, Y, and Z, front to back, left to right, top to bottom. And it seemed like this was preserved into the adult brain. So after publication of this paper, Van got a lot of interesting publicity, including I think a prescient comment that your brain is not a bowl of spaghetti, which was basically the thought of how these things just took the shortest distance, went wherever they wanted. Marco Catani, a very good scientist, however, wrote a reply in Science that in very simple terms said this was rubbish. All he ever saw were maybe 45 or 60 degree crossings. Well, he was using a cue ball tractography that didn't have the resolution of Van's, so didn't really show the detailed fiber crossings, and would average them out when they were a 90-degree crossing to something like this. And of course, what he was showing corresponded to what everybody believed was going on, either artist renditions or simple cartoons of how we all thought fibers went through the brain. Here are some illustrations from a paper I published with Steve Demeter and Gary Van Hoosen. And we had injections in the occipital lobe, frontal lobe, medial temporal lobe, and we showed where they crossed in the dorsal hippocampal commissure, in the inferior forceps of the corpus callosum. And the question then became, well, how do the fibers from these different sites of origin navigate through the white matter? And we had lots of paper drawings of how we thought they went, and published in 1990 these two illustrations, which are an artist's illustration of how we thought the fibers got to the anterior commissure or into the splenium. Interestingly, in one area, we realized they were crossing fibers from the inferior temporal lobe and fibers from the medial occipital lobe, but this was basically an artist's rendition and actually is not true. as I'm going to show you. So we started carefully looking at track tracers. Here's a case like those that Jarrett just showed you, an injection in area 4, primary motor cortex, autoradiography. So these are tritium labeled fibers shown in dark field, where a cord goes down heading toward the corpus callosum, but then another one heads down into the internal capsule. And in fact, these fibers are indeed crossing in an odd way that wasn't really expected. Now, Van went back and looked at Wendell Krieg, an early neuroanatomist who made lesions in the monkey brain here in motor cortex, showing degeneration with the Markey stain that stains degenerating myelin. And this was a reconstruction that Krieg drew, showing fibers making right angle turns as they leave the cortex. Some go into the corpus callosum, but others turn and go down toward the subcortical areas. Well, we wanted to know what was true here. So DMRI fiber reconstructions depend on MRI resolution. They depend on assumptions and restrictions in the reconstruction algorithms, creating the streamlines from the way water diffuses inside axons. And we wanted to know if we could validate these using microscopy and transported labels. So we did a case where we put injections in 4 areas of the monkey cortex. These were 4 different tracers. I'm only going to show you the one that went through area 4, primary motor cortex. This is the central sulcus. It used biotinylated dextranamine, a dextranamine that neurons take up. They transport out their axons and can be immunohistochemically demonstrated with an easy reaction. So here, in fact, is the blackened area where the BDA was injected. And this box shows axons leaving into the centrum semiovale subcortical white matter, some going on toward the corpus callosum. But look, right away, there are fibers already going at right angles to each other. And this higher power, you can see this little, I don't know whether it's a piece of hair or whether it's an axon. I think it's an artifact, but it shows you the orientation that the fibers coming out of the injection somehow have made turns and are going at right angles to it. And if you look at higher power images, what you find here is a BDA labeled axon that branches. Here is a BDA axon that turns at 90 degrees. Here are BDA axons that cross at right angles. Now, the BDA only labeled fibers originating from primary motor cortex. So that's why there's so much here that's unlabeled, okay? But we counted up these examples of turns, crossings, T branches, and I'll show you a little bit of that data later. But let's look a little more detail. Here's the same injection in a section that was counterstained with neutral red, and you can follow the main fibers going out, but others have gone down to terminate in the putamen or up in the dorsal premotor cortex. But with my colleagues in the physics department, they were able to do image recognition to find the labeled fibers and then to color code them according to their orientation. Now, obviously, it's arbitrary where you start. Here's 0 degrees. But again, you can see that many of the fibers cross, and using a method called path probability, they could find the angles, create the outlines, with this being relative proportion of the axons that are going up at 70 degrees or left and right at nearly 180 degrees. And we looked at 3 different cases with injections in primary motor cortex, and they all had basically the same pattern. Now, you might ask, why aren't they identical? It's because the injections are of slightly different sizes and in slightly different parts of primary motor cortex. But it confirmed for us that we were looking at this, and we published a paper, Geometric Navigation of Axons in Cerebral Pathways, comparing DMRI with track tracing and immunohistochemistry. What is that? Well, I'll show you. This is the same area of the white matter, subcortical and in centrum semio-valley under area 4. And these axons are labeled with an antibody called SMI312. It's a panaxonal neurofilament antibody that labels every axon. And you can again see that most of these are going at right angles, but not every one. There are a few that take off at other angles. So this is an image montage of 8 confocal images. Now, if we take those images and do the same thing in part of it with finding the orientation, here's what you get. And the high power again shows you mostly going at right angles, a few heading off at another. And Will Morrison, Gene Stanley, and others in the physics department analyzed this using 3 different ways to see if this was an artifact of computation: autocorrelation, path probability. Pardon me, this is path probability, and I can't see that label, but that one should be a Fourier analysis. Now, that was for all axons. There's another interesting antibody, SMI32, that labels only the axons of large projection neurons from area 4. And here's immunofluorescence of these SMI32 positive axons. Again, you can see they're at right angles. We didn't inject anywhere. All we did is take this section, put it in the SMI32 antibody, which then shows up as red fluorescence, and here's the pattern. Turning that into orientation, we found again mainly right angle, but here's a small population at another angle. These are probably some of those fibers that are heading off like this one, if you can see my arrow here. A few interlopers passing through this grid area. So when we look at SMI312, all fiber orientations, SMI32, just big projections, and area 4 BDA labeled fibers going from rostral to caudal in each of the 3 cases where we put BDA injections into area 4, you again see that the patterns are relatively consistent, although there are differences because the sections will have come from a slightly different part of the white matter, and the injections and or immunohistochemistry will have had some variance. But all in all, pretty compelling. So these axons have to go from primary motor cortex into these different orientations by making turns or branches. So at high power, we could find this is a BDA labeled fiber, and here it is branching and crossing another fiber. This one is in fact making a right angle turn, and it looks like this because it's leaving the plane of section right there. But here's a fiber, the green arrow, showing that it crossed. So we went through and quantified these. What percentage out of a total of 91 axons that we looked at are making right angle turns like this or like that, what percentages are showing T branches of either part? Now, we have a theory that I can't prove to you that during development, fibers branch. And there are developmental studies that show that. But then a branch that doesn't make successful synaptic connections will die back. And it may be that a lot of these right angle turns are left over from branching where one of the branches didn't make excellent connectivity and died back. I'll point out one interesting thing here. This is a Golgi stain. And the Golgi stain in the 1890s created modern neuroscience because it would label, in this case, an entire pyramidal cell with its apical, basal dendrites. You may be able to see the spines, the little spicules where synapses occur. And Santiago Ramon y Cajal figured this out to the point of putting in arrows about how action potentials would connect. But look at this. Here's an axon coming straight down from this pyramidal cell. Here's another one, but look what it does. Right angle branches. This is in gray matter. The Golgi stain doesn't work in white matter because the myelin prevents perfect impregnation. So nobody's ever looked with the Golgi stain in the white matter the way we looked with the tracer injection. Now, here is another tracer injection that Jarrett found. And again, you can see fibers leaving the injection site, but somehow they have turned out here to be at right angles, whereas these, you can imagine, just left the injection site at this right angle. And this isn't just under motor cortex. This is the sagittal stratum in the lateral occipital lobe, which has fibers coming from the geniculocalcarine tract going back to primary visual cortex, fibers from primary visual cortex going forward to association areas, fibers from the corpus callosum crossing. And you may be able to appreciate that there's actually space in here. And one of our questions is, well, what's occupying the space? And my colleague Farzad Mortazavi has been using Clarity to look at three-dimensional axons. This is a section, a thick section from the sagittal stratum. And again, you can see fibers going at two different angles, but you can also find the ones easily now that are coming straight up at you that didn't label in the diffusion MRI. Finally, a couple more slides. This is the rhesus monkey. Van Wadine's diffusion spectrum imaging under primary motor cortex. Fibers crossing the corpus callosum, heading down into the internal capsule. This is higher power, showing the grid structure, and this is the highest power where we can pick up the Z dimension as well as the XY. Is this relevant to what's going on in the human brain? And here is Van's reconstruction from the same imaging approach done in humans at the Martino Center, where you see X and Y and some Z. So we think that this grid structure is in fact ubiquitous in the monkey and ubiquitous in the human. It doesn't mean that some fibers don't stray from it for a variety of reasons that we'd have to go back into development to see. But the question you have to ask is, well, is this fiber coming through? And I'm trying to show one here with my pointer. Does this one branch or not? Is this green fiber going up a branch of this one, or one that was just passing through? If you want to do tractography and follow it to the end, you need to know that. So how do axons traverse the cortical white matter? Do they use traditional smooth arcs, or do they follow the grid? Here's the traditional view, which I hope I've shown you enough data, both with track tracing and DMRI, to say this is not exactly true. You might see this particularly with lower resolution DMRI, where it will take this kind of grid and average it into these curves. But what we need to know is if this fiber enters the grid, does it make a turn to go someplace else? Or does it branch to go someplace else? And right now, our feeling is that in diffusion MRI in humans, you can see the big stems, but you can't follow the individual axons from their origin to their termination through these grid-like structures. And these sharp turns and branches, we measured them, have a radius of less than 15 microns. So they're below the current resolution of even the best DMRI. So what have we learned? Injections of track tracers that label specific fiber populations demonstrate that most cortical tracks conform to grid orientations. 2, immunohistochemical identification of white matter axons, that is, entire populations, not going from X to Y as a tracer would, confirms the predominance of grid structure. And microscopic assessment of this shows they move between grid axes by making right-angle turns or branches on a radius of 15 microns. So what do we need to discover? Is this really, how general is this? We need to do track tracers in other regions and study it with the detail we did of Area 4. We need to determine how many axons entering grid crossings turn or branch there. And we need to create 3D reconstructions of entire pathways as they move across the grid, probably using an approach, a combination of tracer injections and clarity, where we can see millimeter to centimeter thicknesses of the tissue to see exactly where the axons turn and branch. Okay, so any questions if we've got a few minutes? Otherwise, happy to move on and appreciate having the opportunity to talk to you all.
Good. Thank you so much, Doug, for this colorful and wonderful presentation. I have a very quick question before we go to David Perez, who is anxious to start.
I'm not hearing
I
anything.
can see him moving around his head. Hi, David.
Nikos,
So,
I can't hear you.
You're
muted. Nikos, can you hear me?
I have a very quick question, Doug.
Yeah.
Thank you for this colorful and wonderful presentation, fascinating. I have worked with Van before you for several years. So we produced these colors in our first paper, based in '97. But the question I have is if you are familiar with Jeff Lichtman's work, that he works with nano scales. And how, if you are familiar with that work, how do you think it will be insightful to really understand at the nanoscale level what is going on with this kind of geometry. I say that because I remember when I read in a book of Einstein about Brownian motion, he was saying if you are in a very tiny little scale, you will feel how the drops of water are going on a glass in a different way as if you are a human being and you see the drops very, very small. If instead of being a human, you are just a little dust point, you see the whole geometry differently. So that's why I am thinking of Lichtmann, because seeing his images of slices at 0.29 nanometers of tiny little tissues with his amazing new technique, I don't see this geometry, but it's a different scale. I have no idea and no understanding. I just believe that the scale is, and the resolution is extremely important.
I would agree with that. You're talking about Jeff's EM reconstructions.
Yes.
And I will say, you know, Alan Peters was doing electron microscopy from tissue I gave him from my monkeys. And we've looked around, and he, in fact, has a number of images of axons making those kind of T branches, like I showed. Not a lot, not enough that we can publish it or quantify it, but they're there. Now, Lichtmann also tried to use his Brainbow technology, where you go in and label all the different neurons genetically to produce different colors, but those axons couldn't be followed as well as he thought they could. We tried to play with that in the monkey a bit, but got nowhere. So I agree, scale's important, but I don't think scale obscures the fact that we have these grid things.
Wonderful. Thank you so much, Doug. David is next. And we are getting now from amazing neuroanatomy of connectivity to, I would say, mysterious psychiatry, neuropsychiatry. It's in the crossing area of neurology and psychiatry, reviving Freud in, I don't know if I'm correctly saying that, David, but reviving old masters in the new era of neuroscience. Please go ahead, and thank you for being with us.
Nikos, thank you very much for the introduction. I'll say a couple of scene-setting comments in a moment, but let me make sure I can share my screen first. So I'm not familiar with this interface, but let's see what I can do here. I'm going to share my entire screen, and then we'll work through to make sure my slides are visible. So do you all see my slides now? I can't see you all, so I need someone to just speak up and tell me if you see my slides. Nikos, what do you see at your end?
Not yet.
Let's stop sharing. Let's try something else. So how about now? What do you see?
Now I can see your first slide.
Okay, great. So hi, everyone. Nikos, thank you so much for this introduction. Just a couple of background comments. I am both a neurologist and psychiatrist, as well as a research scientist. And my clinical and research efforts are in this entity of functional neurological disorder, really at the origins of modern day neurology and psychiatry. Whereas Nikos was mentioning, in the early history of our fields, Charcot and Freud and many other leaders worked together and learned together side by side. And I'm going to suggest that we're going back to the future, but with some updates. And we'll talk about the updates in terms of our neurobiological understanding of this condition. These are my disclosures. And over the next 20, 25 minutes, I'm hoping to cover two broad areas. One is provide you all a practical work in progress approach to how to understand functional neurologic symptoms, neuropsychiatrically. And let's connect our neuropsychiatric understanding to structural and functional neuroanatomy in this patient population. So you're going to hear me talk about this notion of brain-behavior relationships. And brain-behavior relationships is really the hallmark of behavioral neurology and neuropsychiatry. But what do I mean by behavior? I mean the full spectrum of human behavior. So brain-behavior relationships includes brain-emotion relationships, brain-cognition relationships, brain-perception relationships, and a range of other neuropsychological constructs that you can insert into this framework. Now, in this context, we're going to talk about symptoms, neuropsychological constructs, and how neuropsychological constructs might map onto neurocircuits. And let's begin with the presentation of functional tremor. So here, patients have tremulous movements, and they report that the tremulous movements are out of their control. Okay, I'm shaking, but I don't feel in control of my shaking. How do we understand this? So first, let me show you a video of what we're talking about. This is a patient with a functional tremor. You note a high amplitude postural tremor. Cognitive distraction did not change the tremor. But now we're doing a motor distraction, finger tapping. That postural tremor initially went away, and you're seeing time-locked movements in other instances with the finger tapping. When the finger tapping is very slow, the movements go away entirely. Now with ballistic movements, there's a pause in the tremor. What this video is showing you all is prominent variability, distractibility, and entrainment. The fact that we can influence the rhythm of the tremor based on the rhythm of paced movements in a different body part. In 2024, these are the signs that we use to make a diagnosis of functional tremor. So how do we think about the pathophysiology of this? Well, there was a landmark study in 2009 in Science where a range of individuals were undergoing cortical stimulation while awake in the operating room. And what was observed is that at low cortical stimulation intensities, when stimulating the inferior parietal lobule, as well as the adjacent temporal parietal junction, individuals began to report, I feel like I want to move. And at higher intensity, individuals reported that they moved when no movement had actually occurred. So what insights is this tapping into? Well, it turns out that when we're preparing to move, our sensory motor areas send an afferent copy of the expected sensory consequence of the movement to the inferior parietal lobule. And the inferior parietal lobule is matching the expected sensory consequence to the actual experience that occurs. Our brain predicts before it experiences. So with this in mind, in functional tremor in 2020, there was a landmark study where Mark Hallett and Valerie Voon and colleagues from the NIH identified 8 patients with a functional positional tremor. In position 1, I am shaking and I do not feel in control of my shaking. But in position 2, I can shake and say, yes, I am the owner, I am the author of that shaking. So in this context, they had people go into an fMRI and they compared the activation patterns between functional tremor movements and volitional tremor movements. And what they observed was hypoactivation of the right temporal parietal junction. They also observed alterations in connectivity between the temporal parietal junction and sensory motor areas. So what did the authors say was the potential implication? They argued that this represented a neurosignature or a neural feature of impaired action authorship recognition. So when we think about functional tremor, shaking and not feeling in control of your shaking, the implicated construct may in fact be impaired self-agency perceptions. And neurocircuit-wise, we want to think about an important role for the right temporal parietal junction and its communication to sensory motor areas. So using this framework, let's continue. Now, some patients with functional motor symptoms have major full body shaking events. A subset of those major full body shaking events also have an impairment in speech production and awareness. We call that subset having functional seizures or functional non-epileptic seizures. How do we conceptualize this subset of symptoms? First, let's give you all a preview into what this looks like. Here's a patient admitted to the epilepsy monitoring unit experiencing a functional seizure, also called dissociative seizures. So you saw some major motor bicycling type events, but then the motor movements became more asynchronous, stopping and starting. The rhythmicity of the leg movements, highly variable. You can't see it too well, but the patient's eyes are tightly shut. And throughout the EEG, before the event, during the event, and after, no signs of epileptiform activity. This is what we call a documented functional seizure. So here we saw it with colleagues at the Martino Center, including the study led by Ibai Dez, who's a computational neuroscientist and an assistant professor of radiology at the Martino Center. We employed a relatively novel resting state functional connectivity technique called stepwise functional connectivity. This integrates components of seed-based connectivity as well as graph theory. And what it allows us to do is look at information flow across brain areas. So you can look at information flow, for example, from primary motor areas like the M1 hand, or you could look at information flow bottom up, for example, the sensory portion of the amygdala, specifically the lateral basal amygdala. And what we know in healthy subjects is when you look at information flow from sensory cortices, there's convergent information to multimodal integration brain areas. These multimodal integration brain areas include, in green, the dorsal anterior cingulate, the anterior insula, and the temporal parietal junction. Many of the same brain areas implicated in functional neurologic disorder, including, by the way, when you think about the importance of multimodal integration brain areas, patients with brain, mind, body symptoms, functional neurologic symptoms, have so many different symptoms: tremulous movements, full body shaking, cognitive clouding, widespread body pain, prominent affective symptoms. And these multimodal integration regions, including the anterior insula and the dorsal anterior cingulate, bind not only sensory information, but they're critically important for nociceptive and affective information as well. So it fits really nicely with having a great multiplicity of symptoms in this population. So using this technique of stepwise functional connectivity in 30 patients with FND versus a matched healthy control group, we looked at information flow from primary motor areas and we observed increased information flow to the posterior insula bilaterally in the F&D group versus healthy controls. When we began bottom up in the sensory portion of the amygdala, we observed increased information flow now to the bilateral, left greater than right, anterior insula. So what this suggested to us was a bit of a traffic jam. As you think about crosstalk between motor control areas and core affective processing areas, there seemed to be convergent hyperconnectivity at the level of the insula, anterior and posterior. That was one finding. We also observed that the sensory portion of the amygdala showed increased information flow, or more specifically, increased link step connectivity to the periaqueductal gray. If you can see my cursor, it's a brainstem structure here that I'm highlighting for you all. This PAG area, periaqueductal gray, caught our attention, in part because it's been identified in other task-related affective paradigms in patients with F&D as being hyperactive. And we know that the effector nuclei of the amygdala, the central nucleus, has direct connections to the periaqueductal gray. And this brain structure is involved in fight or flight, freezing, tonic immobility. Right? These observations led us to speculate, and this is speculative, but I think neurobiologically and mechanistically informed, that when we think about these full body shaking events, might the neuropsychological construct be aberrant or abnormal activation of non-conscious primitive defensive behaviors, such as tonic immobility or fight or flight? And might an important circuit in this be amygdala to PAG connectivity? The other thing we've observed is our group has identified and now both discovered and replicated in 2 separate cohorts prominent sensory processing difficulties in this population, including bright lights, loud noises, triggering or amplifying functional neurologic symptoms. This is particularly interesting because there are thalamo-amygdala connections, and might this sensory amygdala to PAG, periaqueductal gray hyperactivity that we observed, help us contextualize why bright lights, loud noises trigger a functional seizure. And you've seen here a very similar demarcation of symptoms, constructs, and circuits. Our initial discovery of some of these neurosignatures, as well as the sensory processing difficulties, are now leading us to interrogate a range of these questions in an entirely new cohort that we're actively collecting. Let's think about another phenomena that we can appreciate clinically. In patients with brain, mind, body symptoms, what we call functional neurologic symptoms, some patients report that strong negative emotion or hyperarousal can trigger or worsen their symptoms as well. How do we put this into context? And here, this brings us back a bit, right? So you heard from Nikos that early formulations of functional neurologic symptoms were framed as conversion disorder. Now, a couple of comments. Conversion disorder and the use of this term has fallen out of favor. It's fallen out of favor because it implies a very specific mechanism to a group of patients that are mechanistically and etiologically heterogeneous. Okay? So we no longer use the term conversion disorder. But we do know from a range of studies, both old and new, that there is relevance to adverse life experiences in this population, including, for example, multiple studies reporting that the burden of childhood maltreatment, physical abuse, sexual abuse in childhood, is associated with increased symptom severity when patients go on to develop a functional neurological disorder. We also know as background that multiple fMRI studies have observed hyperconnectivity between the amygdala and motor control areas, as well as hyperconnectivity between the insula and the dorsal anterior cingulate and motor control areas. So with this in mind, how do we bridge neurocircuitry, disease mechanisms with etiological considerations, including contextualizing how individual differences in childhood maltreatment burden may relate to the pathophysiology of functional neurological disorder? This brings us to our 2021 molecular psychiatry paper, also brilliantly led by Ibai Dez. We used a range of graph theory analyses. I'm going to cover just one, which is link level functional connectivity. And here we looked at connectivity strength pairwise across voxels, whole brain corrected for multiple comparisons. And what we observed in patients with functional neurological disorder is a dose-dependent relationship between the magnitude of childhood physical abuse burden and how closely coupled the amygdala was to primary motor areas, the bilateral precentral gyrus. We also observed the same dose-dependent relationships between the insula and the precentral gyri bilaterally. Now, what made this study particularly notable is it was one of the first studies in the field to include a psychiatric control group. So we recruited patients with mixed depression, anxiety, post-traumatic stress disorder. And it turned out that our psychiatric control group was matched by trauma burden to our functional neurologic disorder group. And yet, for the same magnitude of childhood physical abuse burden, these same dose-dependent relationships in connectivity strength were not observed in our psychiatric controls. And those are the blue dots and lines in the scatter plots there on the screen. We've gone on to speculate that might there be a trauma subtype of functional neurological disorder, whereby individuals with higher rates of post-traumatic stress disorder, higher endorsed rates of physical abuse and sexual abuse, in addition to a range of other childhood maltreatment experiences. Is this a group that's particularly sick? And might this group be particularly refractory to a range of evidence-based treatments? We don't know that latter piece, but we do have quite a bit of evidence, even from our own group, that cross-sectionally, there's a really nice relationship with this subset of individuals endorsing high symptom severity. What's really interesting, too, about this notion of a trauma subtype is in classic psychiatric disease, we know that there's a literature for a trauma subtype of mood, anxiety disorders. People have written about a trauma subtype of substance use disorders. And now in neurologic populations, including migraines, Tourette's, Parkinson's disease, there's now a similar literature to suggest that individuals with those diagnoses who experience childhood maltreatment have higher severity of symptoms related to Parkinson's disease, Tourette's, or migraine. This should challenge you all to think transdiagnostically across a range of neuropsychiatric conditions. Might there be certain neurosignatures or profiles that amplify physical symptoms in this context? More to come, but I think a provocative idea, including it's really important, I think, that we see it cuts across neurologic and psychiatric diagnoses. So here, symptoms being amplified by arousal and negative affect the construct, heightened limbic influence over motor performance, and the implicated circuits being in particular core nodes of the salience network being hyperconnected to sensory motor areas. For those of you less familiar with the salience network, here's a map of this really important network implicated in homeostatic balance. And what you can see here is that the salience network includes the anterior insula, the dorsal anterior cingulate, the dorsal amygdala, the periaqueductal gray, and other subcortical components. Exactly the kinds of brain areas we've been talking about together over the last few minutes. Okay, I'm looking at time, and I want to just very quickly give you a bit of a teaser, so to speak, which is that during the pandemic, my group spent quite a bit of time collaborating with Lisa Feldman Barrett. And she has posited a really groundbreaking theory that is called the theory of constructed emotion. It argues that emotions are not innate. Emotions are not hardwired. In fact, emotions represent acquired concepts, and they represent acquired concepts that are refined over one's life experience. Why did this catch our attention so much in relation to our patient population? Well, patients with functional seizures, for example, report heart racing, shortness of breath, nausea, an internal sense of tremulousness before feeling tremulous on the outside, a whole host of physical symptoms that relate to panic attacks. But what they don't report is the actual experience, emotional experience of panic. This has been called panic attacks without panic. And in this notion that our brain predicts before it actually receives incoming sensory information, we have started to argue that panic without panic may represent, in patients with brain, mind, body disturbances, a form of aberrant emotion construction. What that means is that in a given situation where most of us adaptively would report feeling anxious or stressed out, accompanied by heart racing and shortness of breath and nausea, in this patient population, they are using non-emotion concepts, such as shaking or being unwell, to match to the same incoming sensory information of heart racing, shortness of breath, tremulousness, and nausea. These are entirely, by the way, non-conscious processes, right? So this patient population does not want to be sick. Symptoms are real. They would do their darndest to not experience this. But in the predictive processing framework, things happen very quickly and predictively. And we are wondering if differences in the granularity of emotion concepts and differences in the use of those granular emotion concepts may be important to understand some of the elements involved in functional neurologic symptoms. The other piece here to predictive processing is not just the issuing of predictions, but prediction error learning, right? At times we all issue faulty predictions. The most classic example is walking down the stairs and you anticipate that step being there. It's not. It's very jarring, right? In fact, your body was anticipating all the motor consequences of the step being there, but it just wasn't. And we learn. And it turns out that the salience network is critically important for prediction error learning. Another reason to continue to think about the role of the salience network in the pathophysiology of functional neurological disorder. I'm going to wrap up there, and in the final sort of educational slide, this notion of panic without panic might, what's implicated here, be a feature of aberrant emotion construction. And when we think about the biology of predictive processing, concepts and the issuing of predictions implicates a fair bit the default mode network, and prediction error learning relates to the salience network. I want to thank my team, and here's my contact information. It's really nice to visit with you all. Thank you very much, and thank you, Nikos, for the invitation.
Thank you, David. Thank you so much for this fascinating and dense presentation and extremely important topic. Can you hear me?
I can.
Yeah, it is Darren coming up, so I have just a very quick, extremely quick comment more than a question. I remember when Bernd Cabinet gave a talk in the 1992. You were not around at this time. I was starting at MGH, at Ether Dome. Bernd gave his talk on developmental neurobiology, and Miller Fisher asked him in the very end, Bernd, where is the mind's eye? Now, what comes in my mind for you is, where is all this repressed energy or removed energy that Freud was talking about in the brain, and when it's released, the symptoms go away. But it's more a comment than a question.
I would just factor in this notion of predictive processing, concept generation, concept refinement, and prediction error learning. I think this really cuts across many themes, including brain, mind, body disturbances.
Thank you so much, David.
Thank you, Darren. Good to see you in the background there. Bye, everybody.
Hi, Darren. How are you? So thank you for being with us. Appreciate it very much. Please go ahead. Just remember, in exactly 25 minutes, half hour, we have Maurizio. So I ask everybody to be precise on the time. Thank you so much. Go ahead. We cannot hear you yet.
How do I share my slides in this platform? Oh, I got it. Can't share. Nikos? Oh, I can't hear you. You can hear me. Okay, I can't figure out how to share my slides.
Bring up your slides.
Okay. Hello, can you hear me?
Yes.
Okay. So you should be able to see a...
Oh, I got it.
Okay. There you go.
Okay. Does this show my screen though?
Doesn't show his screen.
Not yet.
Not yet.
Okay.
Yes,
No.
now we can see your screen.
You can see the talk?
We can see a screen, not the top yet.
Okay.
So I believe you should be able to share a particular window rather than the whole screen.
Okay.
Okay, we can see your screen now. Oh. He left the meeting completely. Oh, he's back. Okay, we can see your screen, but not any presentations.
Es que antes a mí me pasaba lo mismo y estaba en un modo raro. No le salía ahí. ¿De acuerdo?
¿Qué pasa?
Okay, I'm going to try to help you let me share my screen. Maybe this way. So
I sent my talk if that might be easier. So I apologize. I seem to be technically challenged here.
The only thing, Darren, is that it's 1 o'clock we have Mauricio, you understand? So
I got you.
so do what you can with the, you don't worry, it's informal meeting, so don't get anxious.
I'm not anxious. I just can't get the slides to work.
All right, no problem.
Podemos mandar la presentación también por correo y la ponemos ahí.
yeah. So let me just share my screen because I'm not sure what you're. So this is what we see when you share your screen, which is this infinite window.
Yeah, I
And
saw that, the infinite window thing.
you should be able to see something like what I'm about to show you, which is, let me just,
Do you have my slides? I emailed them.
Yeah,
you do?
yeah,
Yeah.
yeah.
If you brought them up, I could just say change slides if that might be easier.
El correo.
Okay. So anyway, just in case you can you see my screen?
Yeah, I got to that, and then I hit ignore, and then it just showed my screen infinitely.
Okay, but instead of this Chrome tab thing, you can press on this window tab, and then is when you can see the PowerPoint presentation, and then share.
Where's the window tab?
Well, this is what I see when I try to share my window. So it's right in the middle, right besides Chrome tab. I don't know if you have it configured like that, but this is what I see. So I'll stop sharing now. I don't know if that was of any help.
Okay, we can start with the presentation.
Okay.
Yo puedo ponerle la diapositiva, tal cuando yo tengo que pasar.
Okay, so we can share your presentations, the one you sent us.
Okay.
What you need to do is just say next and
That's perfect. Thank you. And I can go through this quickly enough, but I'd still like to share it with you guys. I'll just do it a little rapidly so that
Okay.
Maurizio can be on time. So thank you so much. I appreciate that. Yay, there it is. I'll get started, guys, for the sake of time, and I really appreciate it, Juan. So I'm a psychiatrist at Massachusetts General Hospital, and at the hospital, I direct the Division of Neurotherapeutics, and that's all surgical and/or device-related treatments for psychiatric illness. So today, what I thought I would talk about would be my work as a physician scientist in developing treatments for OCD. I think you guys are familiar with DBS for OCD, so I'm not going to talk about that. There's also some work of TMS for OCD. In fact, there's an approval of one specific TMS stimulation parameter set by the FDA in the United States. But today, I'm going to talk about transcranial focused ultrasound. Next slide, please.
do you see the next slide?
No.
Oh, no?
Nope. That's all right. I can do it. All good. So OCD is a relatively common disorder, 2 to 3% of the population. The good news is we get half to more than half of patients well with conventional treatments, including medications and cognitive behavioral therapy. But not all patients get well. Next slide, please. There are multiple circuits, cortical striatal-thalamocortical circuits involved in OCD. Main nodes are the orbital frontal cortex, the ventral striatum, and the thalamus, as well as the anterior cingulate cortex. The deep brain stimulation is at the ventral striatal target, for example. So we know that impacting this circuit can help treat OCD. Next slide. I mentioned deep brain stimulation. I think you're familiar with this. It's electrodes placed deep in the brain in a stereotactic manner, with pacemakers on the chest that provide the power and provide the microprocessor for what stimulation to deliver. And currently, the FDA-approved target is down in the lower right. You can see a couple images above of the electrodes in a CT space. But the electrode goes down into the ventral capsule, all the way down to the ventral striatum in the anterior limb of the internal capsule. And you can see that the electrode has multiple contacts that allow you to stimulate in different areas geographically within this space. Next slide. Interoperatively, I saw this yesterday. I was in the OR all day yesterday participating in an implant of DBS for OCD. If you find the right spot, you turn this on and it elicits positive mood instantaneously within seconds. And you turn the electrode off and the positive mood goes away. We do the interoperative stimulation so that we're confident we're in the right place before we cement the electrode in place. Next slide. So that's deep brain stimulation, and you can see that in the middle column. And there's also transcranial magnetic stimulation, which you are all familiar with. The advantage of TMS is that it's non-invasive. The advantage of deep brain stimulation, roughly, is that you can put the electrodes anywhere in the brain, whereas you're limited to the brain surface with TMS. We're excited about focus ultrasound simply because it's both non-invasive, but also allows us to target any brain region. It can target not just the surface of the brain, but anywhere. It has a very good spatial resolution of 2 to 5 millimeters. So much more than TMS, and close, but not quite as focal as deep brain stimulation. It uses a transducer that's placed on the side of the scalp, and different focal length transducers of 80 millimeters, 65 millimeters, 55 millimeters, etc. That's where the focus will be directed into the brain, the energy from the focus ultrasound. Next slide. So what we decided to do is to do a trial in OCD at the ventral capsule ventrostriatal target with focus ultrasound. That's the targets that's approved by the FDA for deep brain stimulation for OCD. However, the hard part is converting what type of mechanical sonication parameters might be equivalent to the electrical stimulation that's delivered with DBS. So before we moved on to treating patients with OCD, we did a study that explored kind of dose finding study in healthy volunteers. We enrolled 12 patients, and 1 week apart, we did 3 sessions in the ventral capsule ventral striatal target, but we also did a stimulation one of the weeks in the entorhinal cortex as an active control site. And you can see the stimulation parameters. We varied things such as the frequency, the duty cycle, and pulse width based on existing data to try to identify which of these protocols, A, B, or C, we would use in order to treat OCD. Next slide. So we put the patients in the scanner to do this. We collected resting bold data before transcranial ultrasound and also had them do mood ratings. We then delivered the transcranial focus ultrasound in the scanner, as you can see in the middle. And you can see the hockey puck-like device, the transducer that delivers the wave with markers on it in the brain image. And then we repeated the resting bold and the ratings of mood. Next slide. So the most important thing, no serious adverse events. It seems safe in all patients, both by asking them in an open label fashion, but also by doing a scale that assesses for adverse events, the safety. Next slide. So excitingly, we found that protocol A made people feel more positive, almost stat sig, but it was only 12 patients. And that protocol C produced more energy. Again, close to stat sig, but only in 12 patients. So these are good initial signals. And we didn't see any acute mood effects with protocol B. Next slide. What we did find, though, is that we were able to alter. I remember we did the pre and post resting bold. We were able to find alterations in resting state functional connectivity with nodes associated with OCD with different stimulation parameters. So you can see in panel A, we find that protocol C increased connectivity between the ventral capsule ventral striatal target and the putamen compared to protocol A and B. In panel B, protocol A increased connectivity between the ventral striatum and the anterior cingulate cortex compared to protocol C. Panel C, VCVS connectivity was increased with the globus pallidus in protocol C. And finally, in D, protocol A increased connectivity between the ventral capsule, ventral striatum, and the orbital frontal cortex. Next slide. So we then went on and have started a study in OCD, and it's the same protocol. The only difference, the baseline and the final TFUS session are the same. But instead of doing one session, we're doing 6 sessions over 2 weeks, with the first and the last being done in the scanner, and the middle 4 being done out of the scanner using the same type of neuronavigation technology we use to do transcranial magnetic stimulation. Next slide. So the first patient could feel the sensations, but tolerated it fine. The second patient couldn't tolerate it at all. They felt a loud, unpleasant buzzing sensation when we stimulated, along with ringing in her ears. Interestingly, though, it started before we even administered TFUS. And she had reported similar headaches and ringing in the ears with loud noises, and also being an MRI in the past. So we're not sure whether the TFUS caused it or the MRI. I'm leaning towards MRI, but we want to be diligent about reporting what happened, given that it started before we even administered TFUS. But we then just removed this patient from the study for just in an abundance of caution. Next slide. So in the one patient we have so far, we've since enrolled 2 more. They did report decreased energy levels, but it felt calm to them. So that was good. This is after 6 sessions. And the feeling, they also had pleasant feeling of drowsiness, and reported that they were sleeping much better than they had for years between the TFUS sessions. Next slide. We also showed widespread decreased resting state functional connectivity between the target that we stimulated, the ventral capsule ventral striatum, and the sites you see here. So these are all decreased connectivity. The putamen, the globus pallidus, the caudate, you can see the thalamus and the anterior cingulate cortex. This gives us some evidence that we are impacting the networks that we want to with the TFUS of the ventral capsule ventral striatum. Next slide. The patient reported the obsessive thoughts were still there. They didn't go away, but the urge to engage in compulsions in response to that was not nearly as strong. And you can see we reached a decrease of their Y-box score post-TFUS. So that's the post, not mid-treatment, but post from baseline, with a 37% reduction in the Y-box. So we were very happy about that. And just for context, 35% decrease in the Y-box is the FDA definition of response. So this person was a responder. We are also following these patients up 1 month, 3 months, and 6 months out. At the last point for this patient at 3 months, they were still improved from baseline, but had lost some of the benefits. So that may be important for us designing durability studies in the future to try to keep gains if they occur in patients. So last slide, in respect for Maurizio, and also to give time for a couple of questions. So far, it looks like with the ventral capsule to ventral striatal TFUS, we can induce acute effects on energy levels, both in healthy individuals and in one patient so far with OCD. We can change connectivity between our target and the circuits that I showed earlier in the talk. And we saw that both in healthy individuals and patients with OCD. And we've also shown that we can reduce Y-box scores, although it's in one patient thus far. So right now we're continuing a study. We're going to enroll 12 patients at Massachusetts General Hospital and 12 with our colleagues at Baylor, so we can get a full sample of open label treatment with TFUS for OCD in this target in 24 patients. So hopefully set the stage to do controlled trials. So with that, I'm going to close my slides and take a couple of seconds for questions, couple of minutes, I mean, for questions, and be ready for Maurizio. So hopefully, Nikos, I got through that fast enough, but also not so fast that you guys didn't get it.
You did a great job, Darren. Thank you so much.
Of course, of course.
Yeah, I mean, and for people to realize how difficult are these interventions, that's the reason of the low number of cases that are reported. And I have just one question.
Yeah.
And basically, what we have discussed for through the years, whether pre-surgical, pre-treatment scanning of diffusion imaging for the fiber tracts, anatomy, etc., would improve this kind of procedures. And how difficult is it? Is it an issue that this is not insured? All this is very costly to do all these pre-surgical things.
Yeah, right now it's a research tool, so that's a very good point, Nikos. I think though, if it ever came to market, the cost would be comparable to TMS, which is good. I do think tractography could help us in the future. You have a resolution at the tip of the energy delivery of about 3 to 5 millimeters. So that's better than the centimeter that you have with TMS, but it's also not as tight as DBS where it's sub-millimeter. So we're going to hit a wider swath of fibers with this than you do with DBS. So it may not be as important, but it could still be helpful, even though it's not doesn't have quite the same resolution, because you could at least to make sure that your 3 to 5 millimeter resolution was within fiber tracts that you want to hit. And lastly, in terms of predicting response, I could also see a day where you administer this to patients before you do DBS at the DBS target, non-invasively, to screen for patients who are more likely to respond to chronic treatment with DBS. That could be a very interesting future application.
Wonderful. Thank you so much, Darren. Thank you.
All right, good to see you guys. Enjoy the Canary Islands. Don't stay in that room too long.
Next year. Take care.
All right, bye-bye.
Bye. So
Zoom.
Zoom.
Okay. Well, let me set it up. It'll take me just a minute.
No grabar eso, ¿eh? So,